Hey, everybody out there, John Marshall for Oncology Unscripted. You might notice that my studio is a little bit more bare today. Well, one of the reasons is that I'm moving. They're kicking me out of this office for a smaller office. That's what you get when you get old. It's discrimination, age discrimination. It's actually appropriate job discrimination. But we've got some blank walls in here, but we've got a lot of bright content. And I am so excited to introduce to you all, if you don't already know him, pretty famous guy in his own rank, Dr. Howard McLeod. Howard and I have known each other for decades. We can say that, I think, with some certainty. And I want him to introduce himself because what we've got to talk about is critical to almost everybody's practice who's listening in. Howard, welcome. Give us a little background on who you are and what your area is.
Oh, thank you, John. It's great to be on your program. I'm a avid listener, first time appearer, as they say. I got drugged into this area of pharmacogenomics, the hard way. A little girl and nearly died of her therapy instead of her leukemia when I was a fellow at St. Jude's, and we ended up finding the gene responsible for that. And then a similar situation when I was over in Glasgow, a man got one dose of 5-fluorouracil and nearly died. And so genetics as a way of predicting toxicity has kind of been the theme of my life going forward. A lot of implementation, a lot of large clinical trials and such. But fast forward to our work at Utah Tech University, a lot of work around what do we do with this? Making it work at Georgetown is one thing. Making it work at a small practice in the wild is a totally different thing. And so how do we implement this at all places as opposed to these others? And the dihydropyrimidine dehydrogenase story that we're going to talk about today is just one example. A lot of activity around supportive care, which pain med, which antiemetic, which antidepressant. But this specific example is very much a toxicity example, and looking forward to discuss it with you.
Well, let's drill into this. So it's one of our themes for this season right now is around the increased utilization of molecular profiling, of identifying sort of positive signals for therapies. And I've always thought that pharmacogenomics, because I work with a guy named John Deacon here, who is, you know John well, and he's been preaching your same gospel for a long time of we need to be doing more pharmacogenomics genetics on our patients to optimize dosing. There's another discussion going on within the cancer community that we're probably overdosing people even to the target, much less to toxicity. And so give me your little, having lived this and having preached this a long time, why don't you think this has had stronger uptake in the community compared to, say, the molecular profiling?
Yeah, and the oncology community is influenced by a couple of key things. One is strong, large, randomized clinical trials. And it's a little bit more difficult to do those kinds of trials, although they have now been done for individualizing classes of therapy. Not individual drugs, but whole areas of especially the supportive care elements. And so that's part of it. The reimbursement needs to be there. There needs to be a way of getting the test in a rapid turnaround. But part of it also is just what are you used to? I remember when PET scans first came out, no one would order one. And finally, radiology got more comfortable calling them. It wasn't just nuclear medicine physicians, but radiologists would now call the PET component. And suddenly, now you visit a cancer patient, you get a CT PET. Maybe not quite that much, but conceptually, it's become so normal. And I think what we're seeing now is a little bit of a tipping point where some of the experience that we've gotten with tumor genomics and with inherited cancer genomics is now causing us to say, wait a minute, I don't really need to waste my time or the patient's, more importantly, giving them the wrong drug when I can weed some things out and choose a better one. And so I think now it's become more of a clinical efficiency tool, and now we're seeing the uptake start to happen.
There have been sort of early adopters on DPD testing, and then later, I think our group here has been later adopters, to be quite blunt. And I will share with you that in the last, I would say almost 10 years, is that when I write that first script for 5-FU or capecitabine for somebody, my sphincters are a little tight for the first couple of weeks because I personally have never written the script to somebody who's been deficient to a life-threatening level. But I've taken care of several patients who've had that diagnosed who then come to us as a tertiary care referral. Start us off, because a lot of our audience probably is not really 100% sure where we are. I'm not asking for your hour-long grand rounds. High level, What are we talking about here? What are we testing for in these patients?
Yeah, so 5-fluorouracil been around for longer than us. It's been around a long time, and most of it is degraded to a less active metabolite or an inactive metabolite that is then eliminated from the body. Dihydropyrimidine dehydrogenase, or DPD, or DPYD is the gene name, is responsible for that first degradation step. And so if that is impaired or deficient even, then a lot more drug can shut down the active pathway, incorporating into DNA, into RNA, inhibiting thymidylate synthase, all the mechanisms that we can guess about how this drug really works. And so you end up with this basically preferential shunting. If you can't get rid of it, then you need to use it. And so it goes down there. And so initially, the cases that have been found by Bob Diazio and by other groups in the Netherlands and other places have really been those cases that have had the extreme toxicity. Death is obviously as extreme as can get, prolonged hospitalization. The case that I first got involved with, which discovered the DPYD STAR2A variant, had 21 days of ICU stay before he survived, and then his oncologist asked for our help to figure out what in the world went wrong.
But how many variants are there then? Are there
Yeah.
innumerable?
Yeah, so there probably are a lot more variants that have not been found yet, but currently the Association of Molecular Pathology, the AMP as it's called, has two tiers of variants that they consider to be essential. The first tier is especially essential, and there's 7 of those. And in the second tier, there's a lot of data, but not as robust, and there's 7 of those. So it's 14 total variants. There have been a number of case reports that have come out even in the last couple months finding new variants. Those need to be vetted and proven to be truly pathogenic, as they say. But the idea is a handful or two, I guess 2 handfuls or maybe 3 handfuls of variants seem to be the ones that cause most of the difficulty. In some populations, so Jay Patel down in Atrium Health in Charlotte has found some variants, and they were an early adopter. They've been routinely applying this. I found some variants that were more commonly found in African American cancer patients. There have been others found more in Southwest Asian patients, Indian, Pakistani. But in general, those 14 seem to capture the bulk of it.
Yeah, I was thinking about the world of Europe, where there was a case, I think, in France, where a patient dies from toxicity, where, correct me if I'm wrong, it's now a law in France, not all the EU, but in France, that you have to measure this before you give 5-FU. Early adopters have said any information is better than none. The later adopters, to your point of we keep finding new variants, is I'm not really sure what to do with the result when I get it back. Does it mean I can't give the drug, or I need to modify it? And if so, how? And so I think that lack of tightness, if you will, is I've been one of my personal struggles. But do you think now we've got enough there to where we should be doing this in everybody?
I do. I'm obviously biased towards the early adoption end of things, but I think we now have an ability to get fairly quick turnaround testing, not same day by any means, but within 3 to 5 days. We can get it's paid for by more and more insurance companies, so that issue is there. And I think what we're seeing is that if you have 2 bad copies, if you're homozygous for variants, that's really an extreme event. And Alan Van Nook at the presentation that he made to the FDA back in January at a public forum there with CR showed some data from a large Mayo study where the homozygous patients were the ones who did extremely poorly. And that was that first black box that happened in January from the FDA. The heterozygotes had a lot of toxicity, hospitalization, et cetera, but they lived. And those in oncology that are my age, I was literally taught you need to nearly kill the patient in order to kill the cancer. And so it's difficult for folks like myself to look and say, well, how can I underdose this? Underdosing is worse than overdosing. But now I think we can refine it where we see, all right, we can do a reduction. If it's an adjuvant patient, the 25 to 50% is the range that's given. If it's an adjuvant patient, I think a 50% reduction, and then going up as tolerated. And that's a key point. Don't neglect that part. If it's a patient receiving active therapy for metastatic disease, then maybe less reduction. It still allows that some patients will get toxicity, but unlikely to be fatal toxicity, and that sort. So I think we're at a point now. Now we are as good as we are now for renal dysfunction or liver dysfunction. So it's far from a perfect world, but we know that this patient needs to be handled slightly differently.
I think
And
that's a really good point. I mean, I was sitting here thinking about my electronic medical record, right, which I love and hate. And, and the nurse that brightly calls me to say, do you know the creatinine is 1.6? And so what do I do? I probably do nothing most of the time, but every now and then I do something. But there is no pop-up in my EMR. Did
right.
you do this testing? Whereas I've got all of these criteria built in for these drugs. And so I'm assuming that's sort of the next evolution as we'll begin to increasingly see that kind of analysis.
Yeah, Dr. Max Smith and Dr. Jay Patel that I've already mentioned. A few others have been at their own institutions have been working hard on that. Right now, it's been places with champions that have been going in there doing the hard work. I think that now that there is an FDA label change, two of them in the last 7 months, and there's now an NCCN guideline change, at least for colorectal cancer, not yet for breast. Now the places that do those more wholesale are going to start doing that. So Epic and Cerner and the others start making big changes. The lookup tables that we all use electronically have now been updated. So it's making it so that someone who doesn't really know how to pronounce dihydropyrimidine dehydrogenase can still benefit from it, because frankly, you don't want to know a lot about that. You just want to know I ordered the test. I mean, there was a day, it was before you and our time, but there was a day when measuring bilirubin was a big deal. It certainly hasn't been for the last 30, 40 years. And there will be a time when, measuring this stuff is just what we do. But right now we're in this transition phase, and we don't want to underdose patients any more than we want to harm them with toxicity. And so finding that, which side do we err on, and going forward is the challenge I think each oncologist is facing.
Remind me what percent of our patients would have some abnormality in one or more of these genes?
It's right around 5% would have some abnormality. The extreme toxicity is very rare. It's less than 1 in 1,000. So you may encounter it in your lifetime. A busy practice like you've had, it, might have come through, but most practices will never see that part. And so the fatal part of it will be caught by doing the testing we're doing now. But really, most of the testing is finding those people that are going to be grade 4 toxicity with hospitalization. And can we at least manage them? There is an approach whereby one knows that this is a heterozygous patient. They have one good copy, one bad copy, and you monitor them more closely and act on it. But the onus is on acting on it. So there is an an antidote available. There's probably an anecdote as well, but there's an antidote available. But it has to be administered in a narrow window. And so the tricky part is, can we orchestrate that? And often it's just easier to start with a dose reduction and then move up in terms of the practical application.
I'm just setting out leading a national platform clinical trial in colorectal cancer where we have not included testing this as an inclusion criteria. Should we add it?
I would, only because it's now in the label.
Yeah.
And so it makes it easier. No, many of the studies will have done DNA collection, and it could be done prospectively accrued but retrospectively tested. That certainly is another viable option. But if the trial has not launched already, then it certainly would be a straightforward part to add. There is a cost to it. There's logistics. But there's a lot of pre-testing going on in a clinical trial anyway, and so one can say, all right, I'll send it all to the same place. LabCorp, Quest, the big labs all do this testing now, so it's not as hard as it was even 5 years ago.
Well, even the molecular profiling companies for their liquid biopsies have started to bundle this in along with
Yes.
germline testing, et cetera. So it's going to be in our faces whether we want it or not if we order these other tests.
This is just the type of thing that we can do to learn more about the patient. We're now at a point where the therapy is so successful that we can care about the toxicity and can care about pain control and all these others. I mean, we've always cared about it, but there's been all this existential piece of saving this patient. Now that therapies are active enough, we can start putting some attention. And we saw what happened in pediatric oncology a couple decades ago, where the survival rates got to the point where they now can worry about cardiac toxicity and such. Breast cancer has had that privilege. Colorectal cancer is now starting to have that privilege, where the therapies are active enough to to now worry about this next layer of issues. So it's an exciting time. Inconvenient that we now need to add more testing, but I think it's an exciting time and a very straightforward way to apply it.
Well, and I think hopefully makes us more successful. Who needs it? What are the right doses and how to move forward and optimize outcomes for our patients is what we're all about. Dr. Howard McLeod, really thank you for jumping on to let me borrow some of your time. I know everyone who's listening in has learned a lot, and probably tomorrow will start changing their practice. Dr. Howard McLeod, thanks a lot.
Wonderful to be here.